now proceed with our next speaker, Dr. John Lewis from the Oisin Biotechnologies. Dr. Lewis.
All right, good evening, everybody. It's a real pleasure to be here at this meeting. I really thank Aubrey and Michael for the invitation to speak. I couldn't really have asked for a better sequence of speakers because James did a fantastic job of covering why senescent cells are important and also why they are such a pain in the ass to work with scientifically. I'm a career academic scientist working in oncology, and I'm relatively new to the senescence field, although I have a lot of experience with killing cells, and that's what I'd like to tell you about today, how Oisin Biotechnologies is working to develop very selective therapies to kill senescent cells in cancer. So I don't really have to give much of a background on what a senescent cell is or what they do. I mean, these are cells that arise through programming in the body, a reaction to outside stresses, oxidative stresses, genotoxic stresses that basically prevent us from developing cancers. And it's really important to note as well that as cells become senescent in the body in response to these stresses, they also can send off factors that can spread, as was fantastically illustrated. And we don't, and I'll reiterate the fact that we don't have great markers for identifying them. So there are some common features that we can use scientifically to identify them in this case. Because of accumulation of active enzymes in lysosomes, we can stain for active beta-galactosidase. But really, these are a very heterogeneous population. And they arise from a variety of different pathways. And I don't want to go through detail about signaling. I just want to highlight the fact that the induction of this cell cycle arrest is mediated through a number of factors: P16, P53, P21. And so we've developed, we've thought about this and thought about, what are the common pathways that we potentially target and create a selective therapy that can start to ablate these cells. And obviously, I don't need to go into the fact that while senescent cells have been implicated in aging and age-related processes, there are also very specific diseases that are also a consequence of aging and other phenotypes that an anti-senescence therapy could address clinically. So it was a really salient point in the last talk about, P16 cells, for instance, may not be the whole story. But you have to look at the data that's been shown in the past few years, and this is what really convinced me that, sure, all senescent cells may not express P16, but it's very clear in a mouse model, if you engineer it so that you can selectively ablate all of the P16 expressing cells, you get dramatic changes in phenotype. And I think, for me, that was very impactful. So in this case, Jan van Dersen's work, if you genetically engineer mice to express a suicide gene driven by a P16 promoter, in this case you use what's called INC-ATTAC. It's a caspase-9, inducible caspase-9 system that allows them to then give a dimerizer that activates apoptosis in cells that are, have activated P16, putatively senescent. And these mice showed, very dramatic changes in their phenotype. So significant improvements in health span, 25% median increase in lifespan, although some heterogeneity, 50% less cancer, and also, functional phenotypes as well. Decreased or delayed cataract formation, decreased frailty, decreased loss of hair. And I think, as a prelude to the next talk, some data that came out recently that really, solidified for me that this was something worth going after as a therapy is Peter de Keyser's work that he published last year, showing using a P53-dependent mechanism, a FOXO4-dependent mechanism, he was able to take in this accelerated aging model mice that are losing their hair, they're becoming frail, and showing treatment, after these phenotypes have already manifested, being able to reverse those phenotypes with an anti-senescence approach. So this to me really solidified the fact that this was a worthwhile development route to take for a therapy. And so that's the basis of Oisin's technology. as a team, we sort of thought about if we're going to develop a therapy that we could potentially use for disease, but also thinking about the general anti-aging community and where this might be used sometime in the future after it's proven clinically. So we wanted to utilize a strategy that's similar, leveraging on successful animal models developed to date. Obviously, we wanted to develop something that has a low toxicity profile, something that's well tolerated, something that can be repeat dosed, and dosed again and again. So something that was non-immunogenic, ideally didn't have overly off-target effects. And obviously, many senescence-related phenotypes are tissue-specific, and the ability to target these drugs to specific tissues would also be a strength. So what I'm going to tell you about today is Oisin's technology that we developed. It's called the Sensolitic Platform. It's a lipid nanoparticle platform that contains a non-integrating DNA plasmid. It's functionalized to produce a chemical inducer of dimerization, or to be activated by a chemical inducer of dimerization that induces a very rapid and irreversible apoptotic response. And probably the most important part of this is the drug delivery system. So the ability to deliver a plasmid DNA systemically to many different tissues without significant toxicity. And I'll be telling you about that in a few moments. So Oisin has developed a platform technology. It's a plasmid-based technology, and contrary to RNAi or delivery of messenger RNA, plasmids are, I love the fact that they can be exquisitely engineered, right, to only be activated in situations where certain pathways are activated, such as P16, P21, or P53. But they can also be engineered with enhancers, repressors, and really exquisitely tuned to specific selective tissues and diseases. So we've created a system and a library of constructs that are active in various circumstances. Sort of the two I'm going to show you data on today are a version of the P16 promoter driving a suicide gene and a version of the P53 promoter driving a suicide gene. And so we built a library of plasmids that are basically a specific selective promoter tied to an ICAS9 inducible suicide gene that's then induced or dimerized through a chemical inducer of dimerization. And so I'll tell you a little bit about many of you may have seen this before, but ICAS9 it's a modified caspase, so it's truncated. The recruitment domain, the card domain, has been chopped off and replaced with an FKBP dimerization domain. These domains interact very strongly with this chemical inducer of dimerization, AP20187, or its clinical analog, AP1903, that's already been shown to be safe in clinical trials phase 2. And so what's really nice about this system is that you transiently express using a plasmid in the target cell. It's only expressed in cells with that pathway active, so P16 or P53 in our case. And basically nothing happens until you add the dimerizer. The small molecule dimerizer is very well tolerated. It goes systemic in a matter of minutes, and it induces an irreversible apoptotic response. Basically, the ICAS9 will then dimerize under these conditions, self-cleave, go to the apoptosome, and carry out a very rapid cell death within 2 or 3 hours. And so it's very hard for cells to escape from this. They can't sort of evolve or get away from it. And so it's definitely final. So some in vitro proof of concept experiments. So utilizing the placental lung myofibroblast cell line, IMR90. In this case, we're inducing senescence using 10 grays of radiation and transfecting cells, in this case with a P16 driving ICAS9. Now ICAS9, because it's a little smaller than caspase-9, can be detected with caspase-9 antibodies, and we see that in cells that haven't been treated with radiation, we don't see any expression of the ICAS9. In cases where they're becoming senescent or they're expressing P16, we see induction of ICAS9, and when we add a little bit of dimerizer to these cells, it's gone. So it's very, very rapidly clearing these cells. And then when we look at the ability of this to actually kill the cells in viability assays, we get basically every cell that's transfected successfully with the plasmid dies. And we've shown through a number of experiments, I'm just showing one example here, if we do a Nexin-5 flow cytometry to look at the sort of the pathway of death, this is confirming that we're actually inducing apoptosis in these cells. Okay, so that's great. So we have a plasmid that's very selective for p16 expressing cells that we can kill them very rapidly upon adding of the dimerizer. The question is, how do we make this into a drug that works in people? So it's really the delivery mechanism that's critical to making this both effective and safe. So we opted to use a lipid nanoparticle platform. Lipid nanoparticles have been used for years. I would say mostly there's been a lot of promise and a lot of investment and sort of very few successes. Now, Alnylam Pharmaceuticals in Boston has just had a recent phase 3 successful trial with an RNAi drug. And the issue is that lipid nanoparticles tend to accumulate in the liver preferentially, and their mechanism of delivering nucleic acids into cells is a positive charge. It's sort of a very simple technology. So they've created lipids that have a positive charge. If you use a sort of a constitutive positive charge, they punch holes in membranes very easily, so they associate and punch holes, disrupt endosomes, disrupt plasma membranes, so you can get stuff into cells really effectively, except they're really toxic, so very low maximum tolerated dose in humans. So in response to this, several companies have developed what's called a conditionally cationic lipid. So this is a lipid that's generally neutral in the bloodstream, gets taken up into endosomes, becomes cationic in that acidic environment. And so these are sort of the subject of the current clinical programs that are sort of making their way through clinical trials for lipid nanoparticles. And they work, but they're still quite toxic. So so the ideal delivery system is a delivery system that can use neutral lipids that are non-toxic, but use an alternative mechanism of intracellular delivery of nucleic acids. So I'm going to give you a tiny bit of background as to how we got to this point. So if you look at, so you have a lipid nanoparticle, it has to get inside a cell. It's got to get through an intact plasma membrane with all its defenses. So viruses have evolved, over millions of years to be able to solve this problem. So envelope viruses have evolved a variety of fusion proteins. Unfortunately, as you can see, these fusion proteins are beautiful and gigantic and elegant, and the way that they bring membranes together and create pores and mix lipids is really, really fantastic. The problem is to attach this to a lipid nanoparticle is insane because they're multi-protein, multi-subunit. They have these gigantic ectodomains that are highly immunogenic. So fortunately, there's a Canadian researcher, who's been studying all his life these fusogenic orthoreoviruses. And what he discovered in this particular class of viruses, that they don't use a fusion protein to enter cells, but once they enter cells in their reptilian or bird hosts, they cause all of the cells around them to rapidly fuse together. And so he spent his career characterizing this class of fusion-associated small transmembrane proteins that are 2 orders of magnitude smaller than the smallest fusion protein produced by any other virus. But they are sufficient to induce cell-to-cell fusion, and most importantly, lipid nanoparticle-to-cell fusion. And so, so if you incorporate these proteins into a lipid nanoparticle platform, it's a neutral lipid nanoparticle platform, you'll find that neutral lipids by themselves are extremely poor at delivering things. So in this case, we're delivering an M-cherry plasmid to cancer cells. And so without the fusogenic protein in there, you get no delivery. With it, you get fantastic delivery. So it increases delivery of a neutral lipid formulation by 80 to 350 times. So, and these are well tolerated in vivo. So this is just an example. We're delivering an mRNA expressing luciferase, injecting in the tail vein. We get systemic expression of luciferase throughout the body. You get some accumulation in the lungs and the liver, but we get a broad expression in many tissues, including skin and soft tissues throughout the body. And so this platform then is what we're using then to deliver this anti-senescent cell payload. And so the platform's called Fusegenics. It uses a neutral lipid formulation that's non-toxic and well tolerated, but it uses these fusogenic proteins to deliver intracellularly. So I'm not going to go into all the data, but we've done, it actually took us 3 years to create an antibody against these proteins. They're really not immunogenic whatsoever. And the reason for this is because most of it is a transmembrane domain. They're really lipophilic, so they sort of pack lipids around them, and they have a very low profile to the immune system. And so we spent a lot of time working on these, engineering these fusogenic proteins to make them better. I'm not going to get into it all, but we're at the point now where we have a scalable manufacturing platform to create these at scale, lyophilize them even, and ship them around for use. So let me show you data now, taking the P16 activated caspase-9 and putting it in vivo in mice. So in this case, we've done an experiment now with 16 mice. These are an aged mouse cohort, so 80 weeks old. We divided them into 3 groups where we're giving them a control LNP. We're actually not giving them the dimerizer, or 2 doses, 5 and 10 mg per kg. And 10 mg per kg, if you know this field, is quite a huge dose. So what we did is we treated these animals a single time, so tail vein injection. We waited 96 hours, and then we gave them a single dose of dimerizer, also IV. And then we waited 2 more days, and then we collected tissues, blood, and in this case we're doing a sensitive RT-PCR and controlling it with some housekeeping genes, and we get a convincing dose-dependent reduction in P16 expression in a variety of tissues. I'm going to show a couple images where we spent a lot of time optimizing Betagal staining in mice. These are the prettiest images we got, but we saw in multiple tissues, we saw a dose-dependent reduction in the expression of beta-galactosidase as well. So very, very encouraging data. As far as, obviously if creating data in the lab is great, but if we're going to translate this into humans, there's a lot of things that must be figured out. Toxicology is extremely important. It's important for a drug that you're going to deliver more than once to make sure that you don't create any neutralizing antibodies. So we've done a ton of studies looking at repeat dosing, and we don't produce any anti-drug antibodies whatsoever, so we can give this in repeated doses over time without any reduction in efficacy. CARPA assays are up there. So CARPA is something I learned about recently. It's an immunohypersensitivity, so it's a complement-activated immunohypersensitivity response that actually many patients who receive nanoparticle therapies like Doxil, which is a chemotherapy, can have. And so we've run all the assays for this, and it's got a lower profile than Doxil, so it's very well tolerated that way. And I'm happy to say we've done some pilot non-human primate studies giving 10 times the maximum estimated human dose, and it was extremely well tolerated as well. And so we're just sort of going through that information. So those monkeys actually got the treatment, both the P53 and P16 treatment alone and in combination, and the dimerizer. So we're going to be able to look at that and get some rich data. We're in the process of working through that now. I'll keep coming back to this point. So the tolerability of these formulations is really important, and I always show this slide because it shows you sort of all the efforts that have been put into clinical trials of lipid nanoparticles and why they failed. So if you look at the first 3 programs, these were really promising 10 years ago, using cationic liposomes and lipid nanoparticles, and if you can see by their maximum tolerated dose, way below 1 mg per kg, these programs all failed due to liver-related toxicity. So the second generation in the middle, conditionally cationic lipids, SNALPs, some of them call them, SMARTicles is a crazy name. So these were tolerated to more or less better extent, and some of those programs have been successful and will result in approved drugs, but all of the targets are liver. Because the nanoparticles preferentially accumulate in the liver, you're going to see dose-limiting toxicity if you don't use a neutral lipid formulation. And you can see Anil Sud at MD Anderson working on an RNAi target, FA2, did a phase 1 using a DOPC, a neutral lipid formulation similar to ours. And so he wasn't able to find a maximum tolerated dose in his phase 1 study. So based on our rat and non-human primate studies, we expect our formulation to be equally as well tolerated. And yeah, so I don't know how much time I have left, but I'd like to tell you a little mini story after this on cancer, because I promised I would. So we're currently evaluating sort of a variety of different constructs to see which one is the best to bring into humans. And obviously it's been talked about at this conference, incorporation of biomarkers that are viable endpoints for clinical trials and also viable in animal models to look at efficacy. So we're having these conversations at this meeting, and we're keen to talk to anybody who has a great biomarker. Obviously we have cohorts of mice now looking at lifespan and health span of these mice. And thinking about the transition to the clinical stage where we're getting GMP manufacturing and doing our GLP tox analysis. So I'm going to switch to cancer for a second because this is our route to the clinic. So my day job is a prostate cancer researcher. The one thing that really intrigued me about the crossover between senescence and cancer is the activation of the p53 pathway. p53, as you've heard already, is the most mutated gene in cancer. And what's interesting, so there's a lot of cancers that have a high burden of p53 mutations. I put prostate up there because it's actually relatively low. Average might be 10. Vast majority of prostate cancers are low grade. Once you get to metastatic disease, that mutation rate is well over 50%. So this is a viable target for cancers. And while p53 protein itself hasn't been a great target for oncology therapies, I think the pathway is great. Because if you think about p53, you can get sort of two kinds of mutations. Cells will, with all the stress that cells have through replication and mutation burden, so they'll activate p53 to either resolve the damage or to go through apoptosis. So cells either mutate or get rid of p53 to get around this. And so as a result, the actual activation pathways which are driving this are highly upregulated. So we thought, can we exploit this, this activation, transactivation to kill cancer cells? And so I'm skipping over all the in vitro data because I only have 3 slides left. I want to show you some in vivo data because that's what's really important. So in this case, we're very similar formulation to what I showed you before. So there's a p53, engineered p53 promoter driving this ICAS9 suicide gene wrapped in a lipid nanoparticle, neutral lipid nanoparticle. In this case, we're growing gigantic prostate cancer tumors in an immunocompromised mouse. These are non-skid mice. So we're growing them up to, in this case, up to over 500 millimeter cubes, so big tumors. We're doing a single intratumoral injection of the nanoparticle, waiting 3 days, and then doing a systemic injection of the dimerizer. Incredible. So we saw basically most of the tumors reduced 90 to 95% in 48 hours. And this is not amazing for an intratumoral injection, but I was very pleased because this means we're successfully transfecting the plasmid into the majority of the tumor cells, which I thought was very exciting. So the real proof in the pudding is to be able to do it with a systemic injection. So we've done those studies. This is just an example of 4 mice in that cohort. So these are mice. We've grown the same very large tumors, growing them to a size of 500 millimeter cubed. In this case, we're giving 4 daily tail vein injections of the LNP, and on the 5th day we're giving them a single dose of the dimerizer systemically, and again we just saw remarkable results, so between 50 and 98% tumor reduction in just 2 days, and I'm almost at the last slide. And so this resulted in a significant prolongation of survival with a single dose in these animals, and on average we did 6 mice per group, almost a 70% reduction in tumor volume. So one thing that really important as well, primary tumors don't kill patients in most situations. It's the metastatic disease. So we're really interested in seeing whether we can hit metastatic cancers. Obviously, this will be the population we go into for first clinical trials as well. So we've done a number of models looking at the ability of these LNPs to control metastatic disease. In this case, we've got a prostate cancer systemic metastasis model, and in this case as well, an immunocompetent melanoma model, obviously, which is important as well. And in both cases, with a multiple dosing regimen, we're able to control this disease effectively. So we're on the path. So obviously, the long-term goal of Oisin is to develop, senescence clearing therapies for aging and age-related diseases. But I think in the short term, it's really important, not only for the nanoparticle technology, but also for this platform technology to prove it in the clinic, safety and efficacy. So we've already got a phase 1, phase 2b trial designed. And we're right now gearing up for our GLP talks that'll be enabling for those studies. We're hoping to dose our first person in early 2019. And we're excited about sort of these adaptive design studies and be able to accelerate the translation of this technology. One thing I'll mention as well, so there are many cancers that this could work in. In Canada, we can actually do a phase 1 trial with all comers basically, so colorectal, prostate, lung, et cetera. And so we'll be looking for the biggest signal in the most important cancer to be able to expand that cohort and do the phase 2. So with that, I'll stop and take your questions. Obviously, this project, there's a lot of supporters here in the room, Methaluza Foundation, Kazoo, Sens Research Foundation, and many private angel investors. This is sort of a work of passion by the founders, which has been very exciting to be a part of. And not only do we have a great team at Oisin, but supported by a large, capable lab that actually does all the work, and some great scientists. So thank you very much.
Thank you.